people come in this room?
Let me check.
Well, there's 7 attendees right now. You can just look at the participant list down.
13, I think.
Yeah, well, there's 6 of us are panelists.
Yeah, there's quite a few already.
For your information,
and we're live on YouTube.
My student says the live stream on the KNAW talk on YouTube isn't working. I'll ask her still not.
I just hit go live, so it should be online now. I just heard that the stream is working, so it should be fine now.
Okay, thanks. Okay, so oh. I hope that the YouTube stream is working. I get mixed messages, but I think we'll wait another 1 or 2 minutes and then we'll start. Yeah, exactly. Yeah, so Chase, also Bela says that YouTube hangs. And I got this from other people. So it loops for 30 seconds and it doesn't work.
Well, there might be 2 streams because we did one test session and that one might be the one that hangs. And there should also be one that's working well. But I'll double check.
And is it clear for people which one they have to pick?
Maybe I removed the old one.
Maybe that's Sebastian. Yeah, okay. Okay, so the good news is that I actually received exactly that information from someone, that there's 2 and that 1 works. So if you remove the bad one, then I think we should all be clear. Let me know if we're ready to go, Chase.
Lidwij, I deleted the old one. So I think we should only have one operational one at the moment.
Okay, so you say we're ready to go. Okay, good. Well, okay, I see that all thumbs up, so I think we're ready to go. So let me start by welcoming all of you very warmly to this first session of our KNAW BiophysicsNL webinars. let me first introduce our co-organizers. This is organized by Chase, who is sitting there with the white shirts. Chase Broeders from the Free University. There's Greg Stevens from the Free University, and there's Tom Shimizu from the AMOF and the Free University, and myself. I'm Liedewijn Laan. I'm from TU Delft, Technical University of Delft. This is the first session of our online version of a very sort of classical series of meetings that we've been having in the Netherlands. So basically we have had it for almost 20 years. We've had meetings where we meet as a community in a very old building. It's a Royal Dutch Academy of Sciences building, and we discuss our research together. And obviously this has not been able to go on, so we thought we should find some new formats. And I have to say, I'm personally very pleased with how we set it up now and which speakers we got. So I'm excited that now we're going to have talks by Richard Neher, who's going to talk about tracking of viruses. I mean, what is more timely than that? And also Kristina Ganzinger, who works about information processes in synthetic cells. So I guess also that's kind of a big topic in the Netherlands right now. So before I tell you more about the speakers, let me tell you a bit about the procedure. So what's going to be, we're going to have 2 talks, both are 30 minutes. And during the talk, you can basically write questions in the chat, and then afterwards they will be posted to the speakers. After the first 2 talks are over, we switch to a tutorial session where now people that have registered for the tutorial, they will be part of the panel. So you should see them like you see us, and they can ask questions during the tutorial, which will be more technical. But this can also be followed on YouTube. So you can choose whether you want to go on into the tutorial or you actually go to YouTube. I think that was all the sort of practical things I had to announce. I don't see any worried faces. I think I said everything. Good. So let me start by introducing Richard Neher. So Richard Neher is actually a theoretical physicist who did his PhD in Munich. He worked with Ulrich Gerland, I think. Let's see if I get this correctly. In Munich, and afterwards went to the KATP in Santa Barbara to do his continuous research. Then he worked for a while in, I think, Tübingen, and now he is having his own group in Basel, working mostly on evolutionary topics. And I guess without any further ado, let's start and hear what Richard has to say about COVID-19. Thank you, Richard.
Yeah, thank you very much for having me. It's a pleasure to present sort of what we've been doing on the pandemic over the last few months, and it's it's an honor to be here as the lead speaker at this online reinstallment of that lecture series. So let me share my screen. So you should be able to see my slides now. These slides are actually available as HTML online on our lab webpage. In case you want to go back for references or so forth, you find them in the tab with all the talks. And yeah, what I was hoping to do here in these sort of 20 something minutes of the general audience talk is to recapitulate a little bit how we've been using genome sequences to track the spread of SARS-CoV-2 out of Wuhan across the world, and give you a sense of what we can use the genomes for in our response to the pandemic. And in the tutorial session, I'll go a little bit more into the modeling of adaptive virus evolution, more in the context of influenza, where we're not so much operating on a kind of in an emergency mode, but where the whole thing is sort of more settled, and there is kind of serious modeling and theory, where going into understanding the evolution of influenza is not so much just trying to get on top of things as it is with SARS-CoV-2. But let's sort of start at the beginning, and probably very few of you took note of these kind of news articles that popped up end of December, first days of January in from China, and we're usually sort of on the later pages of a newspaper or somewhere down there on a news webpage, alerting the world that yes, there has been sort of a mysterious virus causing severe pneumonia in Wuhan, And at the time, nobody really knew what it was. People kind of thought, probably people, some animals had some virus and infected a few people, but there was no evidence yet, at least known to the world, that this virus was spreading from human to human and had the potential to cause a pandemic. But then things sort of actually started to change very quickly. Sort of after this initial announcement of these cases that are linked to the fish market was closed, and just a few days later, and that is really really sort of an unprecedented scientific effort. Just about 2 weeks later, Chinese scientists in Wuhan, they had already deciphered the entire genome of this virus and posted it publicly for others to analyze. And that, for example, was the crucial information for the first PCR diagnostic tests to be developed, which meant that we could identify and track this virus globally from the very beginning, from mid-January on, in hundreds of labs around the world. And truth be told, just a few days later, we had the first cases identified in Thailand and Japan. They put sort of the entire city of Wuhan under quarantine towards the end of January. And we had the first cases in Europe also towards the end of January. So here I list some cluster of cases that was identified in Germany around the 28th of January. And by that point in time, the virus probably was pretty much everywhere, and just wasn't sort of discovered much at the time. So what are we actually dealing with? So what I'm showing here in this graph is a phylogenetic tree. So phylogenetic trees, for those who don't work on these topics, phylogenetic trees are a bit of a family tree. They show you the relationships of the evolutionary closeness of different, in this case, viruses, but could be animals, could be plants, whatnot. And so the way they are arranged here is that the past is on the left, and the samples that we are looking at are on the right. The vertical distance here doesn't really have much of a meaning. It's just for illustration purposes. And the distance between any two such tips is kind of the distance you go to the common ancestor and then back to the tip. What is shown here is a phylogenetic tree of so-called beta coronaviruses, which are a large family of viruses, and some of these representative from that family infect humans. And obviously that's the family where SARS-CoV-2 resides in, and it has a relative, not a close relative, but also not a very distant relative, but that's SARS-CoV-1, which is the SARS virus that some 15, 20 years ago caused the SARS outbreak in Asia. But then there's also 3 other viruses in this family that infect humans. And those would be, for once, the so-called seasonal coronaviruses, which are common cold virus. These viruses, they typically return every winter and cause sort of cough and runny noses, but no severe disease. And then there is MERS, which stands for Middle Eastern Respiratory Syndrome virus, which is a virus that was discovered a couple of years ago in the Middle Eastern region and sort of linked to repeated transmissions from camels to humans. So this is a virus that's circulating in camels and sort of spills over to humans sort of quite often, but never really transmits much in humans ongoing. So that is just to illustrate that there's quite some diversity here in the beta coronaviruses. We have a virus that is common in a certain type of animal and often spills over, but doesn't sort of cause outbreaks in humans. We have 2 viruses which circulate endemically in humans at large numbers coming back every winter, but not really causing much severe disease. MERS is actually quite serious for humans, so it's a very high case fatality ratio. And then there's the 2 SARS viruses, which seems to spread very well among humans, and are also quite cause severe disease, at least in a significant fraction of the population. Now, zooming into the SARS part of the tree a little bit, we have SARS-CoV-1, so the original SARS up here, and SARS-CoV-2 down here, in blue being the different being isolates from animals, mostly bats. So the coronaviruses, they circulate with high diversity in bat species, but also in pangolins. And this was a discussion that was sort of quite heated towards the beginning of the pandemic. Where did this virus come from? What kind of cute animal sort of brought it to humans? it's still not really settled. The closest relative of SARS-CoV-2 that we have is this sort of bat isolate here, which was sampled in Yunnan in southern China and has the abbreviation RATG13. Its distance from SARS-CoV-2 is about is such that through random mutation, about 4% of the nucleotides have changed between these two. So they're 96% identical at the genomic level. And this distance or this similarity suggests that they had a common ancestor a few decades ago. So this is not an immediate common ancestor, immediate sort of ancestor of the current outbreak, but it's sort of a pretty close relative on a time scale of a few decades. And what I'll talk about in the remainder of the talk is actually zooming into the red bit here and trying to use mutations that happen as SARS-CoV-2 transmits among humans to track how it's spreading. And we do that in NextStrain. So NextStrain is a project that me and Trevor Bedford from Seattle, the Free Touch in Seattle, have been running for a couple of years now. It started with efforts to track influenza, and I'll talk a little bit more about the influenza part in a minute when I get to the second part of the talk. But we've been using this platform to track Ebola, Zika, and all sorts of other viruses. So when SARS-CoV-2 appeared in the horizon in January, we were sort of ready to go and provide an analysis of this virus to the world online, on the internet, updated once a day or multiple times a day, depending on how the data comes in. And it has sort of received an enormous amount of attention in the course of this pandemic, to the point that we had sort of hundreds of thousands of visitors on the site a day. So it's died down a little bit now, but it's still sort of multiple, tens of thousands that visit the site every week. So in the very beginning, there wasn't actually much data to shake a stick at. So this is the data on January 26. And I'll just talk you through this tree a little bit, which doesn't really look like much of a tree, to be perfectly honest, because there's just so little diversity. So what you have here at the very left have genomes that are identical to this prototypical original Wuhan strain. So everything that's here on the left with 0 mutations is on the entire 30,000 genome, in the entire 30,000 base genome, there's no difference. And then you have a few that are sort of 1 mutation away. There's one cluster which is actually 3, 4, 5 mutations away, and a few one-offs with 1, 2 mutations. So this is kind of what you'd expect if this virus had a very, very recent common ancestor, meaning that there was a singular event that brought it from animals to humans, right? It's not that there was a diverse population in animals and they spilled over multiple times to humans. There was a singular event, and from then on, it spread effectively in humans. And that was one of the central questions that people wanted to ask at the very beginning of the pandemic. What it looked like there was human-to-human spread, it was the genomes that sort of unambiguously told this story. Since then, there has been sort of an unprecedented effort of sequencing this virus, and we've never seen anything like it before. So this graph is now a few weeks old. We are currently at about 130,000 full genomes, which is ultimately a substantial fraction of all the confirmed cases. And it has to be said that over half of all genomes come from the UK, because they have gone absolutely bonkers sequencing whatever they get their hands on. There's a huge variance in how much different people sequence. So the Netherlands actually quite good. They sequence a lot of genomes, but not nearly as many as the UK, but certainly many more as say sort of Germany would. And yeah, the scale of this graph is and also the analysis that it entails for us is a little bit daunting. But let me just point out a few features that I think are important. So here at the beginning, it was a fairly slow rise, which is to be expected because there weren't actually that many cases, or actually many cases known up to that point. And then in March, we know the big outbreaks, they started end of February across Europe. And what you have here in this magenta color, that is the sampling date of a particular genome. So that's the date when somebody got sort of the swab sort of stuck up his nose or her nose. And then the turquoise dot to the right of this, that is the date when this genome was publicly available, right? Available to be analyzed to understand the pandemic. And in many cases, we have a very, very brief lag, only about a week, 2 weeks, 3 weeks between the sample being taken and the sequence being available. Of course, there's also a lot of backfill, like all the stuff here in the lower right corner. That is genomes that were released in August, but which are samples from March, which are nice to have. But what is really important is that there is sort of a pretty fair chunk of genomes that are available to be analyzed just a few days to weeks after the sample is taken, which gives us this kind of real-time insight into how this virus is spreading. So I showed you this tiny little tree before. So this is how this looked like at some point over the summer. It's not the most up-to-date figure, but you get the idea. And so this here at the very right is again sort of the Wuhan strains. And you see in this sort of discrete, discrete stripes, how one mutation accumulated after the next. And sort of nowish, most of these genomes that we sample are some 10, 15 mutations away from the original strain. There's lots and lots of different lineages. So there's many kind of private mutations that are super rare. There's comparatively few mutations that are common among many strains. One such mutation would, for example, be mutations that sort of fall on these branches here, from which a large chunk of the future population descends. But the bulk of mutations are actually very rare and kind of private to many strains. But another thing to point out that the majority, the absolute great majority of these mutations are sort of completely irrelevant from a functional point of view. These are just copying mistakes that the virus happens to make, and they don't screw things up too badly, so they keep circulating. And then there's sort of the rare mutation every once in a while that we think, this might actually change the properties of the virus in some way that it enhances transmission or so forth. And there's one such mutation that I'll come back to later on. But it is important to realize that almost all of these mutations are completely meaningless from a functional point of view. And if you see headlines like, well, the virus is mutating, everybody run, that is usually not sort of substantiated in evidence, but sort of just a clickbait by the media. Good, but there is sort of a very systematic pattern in this mutations. For example, if you now sort of plot all these genomes as a function of date on the x-axis and number of mutations on the y-axis, you see sort of a pretty nice sort of consistent increase in the number of mutations with a rate of about 2 per week. It's not that this is sort of perfectly Poissonian mutation stats. They're quite over dispersed. So sometimes you get 3 in a row and sometimes none for 8 weeks. But sort of on average, you find sort of one mutation every 2-3 weeks or so. And this gives us this allows us to dig into how the virus is spreading, because a mutation is going to be inherited by sort of all future descendants of a particular transmission chain, giving us, providing structure and sort of extra information on the transmission process. So this is how sort of a tree sort of looks these days, and sort of the rough breakdown of different variants globally. So this tree here, as you notice now, is on a time scale. So we have kind of warped using that linear relation between the number of mutations and time, you sort of warp the branch lengths such that every node gets to lie at a position where it was sampled. And what you can see here, we've sort of defined sort of various broad categories of genetic groups, so we can track a little bit sort of how common where. And what you can see is that in Asia in particular, we had sort of the initial dominance of these variants 19A, 19B, which then subsequently were sort of displaced more or less by the descendants of this 20A clade, which is what seeded the big Italian outbreak and subsequently lots of other European outbreak, and then the North American outbreak, which also then completely dominated the South American outbreak. So, and this is one mutation that I already pointed out earlier. It's sort of a mutation that happened in the spike protein. The spike protein is the surface protein of the virus, and so you'd expect, a priori that the surface protein of the virus would be a target where the virus has room to improve, because it comes from bats and or some other animal we don't actually know. And sort of the surface receptors that the virus uses might have sort of different properties in different animals. So there is sort of some scope for adaptation, for optimization of this virus to humans. And this is what people at Los Alamos at the time sort of observed and sort of tried to substantiate, that a mutation that happened in this part of the tree in the protein spike at position 614 from D to G, that this enhanced the transmissibility of the virus by sort of possibly by stabilizing the spike protein trimer on the viral surface. And yeah, what you see is that these initial genomes in clades 19A, 19B, which dominated China, and so Singapore and sort of various other places in Asia, they didn't have that mutation. And over time, they were sort of displaced by viruses that do have this mutation, which is, looks at the face of it like sort of a pretty clean example of sort of a selective sweep. But one has to take this with a slight grain of salt, because you have very strong founder effects here. This virus got into a very susceptible population in Europe, and then whoever gets in first makes a big outbreak and ends up dominating, whether this is an actual transmission advantage or not. But careful analysis and also cell culture analysis, experimental analysis, do suggest that this mutation actually does make a difference and enhances transmission. Okay, so the next few minutes I wanted to spend on some sort of very much more, what I just showed you was sort of a very broad, big picture, how does the virus change on a global scale? And now I want to spend a few minutes on discussing how this virus is, how we can use these sequences to track things that happen at a very local scale. And what I've sketched here is sort of a transmission tree, with every red dot being an infection of a person. Then there's some small fraction of cases here in blue, but we actually sample and sequence the genome of. And as the virus transmits, it once in a while sort of has a mutation, which are these little diamonds here, which are then inherited by everybody subsequently in that transmission chain. We now have these genomes here on the right, which are sort of indicated by black bars with sort of colored diamonds as mutations. And these genomes are what we can use to reconstruct the tree process, right? So this is exactly what happens. You group them by similarity. And in this toy example, it's sort of quite straightforward to come up with this little toy tree, which it has similarities to the actual transmission process, which we are not observing, but sort of lacks resolution in many cases, right? But we can still use this, the reconstructed tree here as a guide to tell us which part of the outbreak belong together, what are clusters, what are not clusters, and thereby sort of focus and make more efficient this sort of track and trace contact tracing system that is in place, right? If you have two genomes that are very different, there's no point looking for a common contact. If you have a bunch of sequences in a city and they're all exactly identical, you might want to up your efforts and find that super spreading event, right? So it's basically a complement to contact tracing, in that it is so sensitive on a time scale of greater 2 weeks, 4 weeks, while contact tracing is sort of sensitive on a time scale of a week or maybe max 2, because who knows, you've been to dinner with like 3 weeks ago, right? So it's operating on complementary time scale, and thereby, if used appropriately, can be quite informative and complementary to the effort. That said, given the high case numbers right now, there's probably not a lot of that you can do with contact tracing anyway. But I was just going to, my colleague Tanja Stadler here at the ETH, she has sort of launched a very extensive effort of sequencing, everything that's going on in Switzerland. And what you see here is sort of a tree with the sequences from the first wave back in March, and then sort of all these little descendants, lots and lots of different clusters that all sort of connect back to the first wave and tell you how sort of many independent variants have been going forward, how many transmission chains were maintained over the summer, and sort of fuel the resurgence, the second wave now. And there's sort of one cluster that I just wanted to zoom into, which also has a bunch of sequences from the Netherlands and Switzerland and the UK. And that's possibly a cluster that is sort of linked to some common holiday destination, where sort of people from Northern and Central Europe went over the summer, picked it up there, brought it back home. We don't actually know where this one is, but it's sort of one example where by combining sequences from different countries in Europe, you get sort of some sense of how the virus over summered and then sort of now comes back as we head into winter. Good. So I'm almost at the end. There was, and I guess, we started a few minutes late, so I probably should wrap up, but no immediate cause for alarm.
No, you can take maybe 2 or 3 more minutes if you like.
Okay, very good. Yeah, so I just wanted to, say a few words on, and maybe the slide is a little bit outdated now and shouldn't be sort of too much to what's next, sort of, but what's going on now. So if you look at influenza cases, this is data from the US over the last 20 years, you see very pronounced winter peaks, right? And we all know that we get colds in winter, we get flu in winter, and so forth, right? So there is seasonality in many respiratory viruses, and there's little reason to expect that SARS-CoV-2 is any different because it transmits along the same pathways. It has sort of a virus that has similar characteristics. It's sort of an envelope virus that sort of needs to get to epithelial cells in your airways. And people often argue that this happens for a seasonal an endemic virus that comes back every year, but for a pandemic this is not so important because in the pandemic, there is a huge number of people that are susceptible. This virus can spread summer as it can spread in winter. Which is true, but there's still sort of every reason to believe that this virus does spread better in winter than it does in summer. And previous pandemics, for example, the 2009 swine flu pandemic, which is the yellow part here, which kind of disrupts this pattern in a very prominent way, that sort of showed pretty similar patterns to what we see now. So if you look at data from 2009 for influenza, you see here broken down into northern US and southern US, you see that the northern US had sort of a double peak pattern, while the southern US had sort of one big, long, drawn out, not as intense, but sort of never stopping kind of pandemic curve. And it's pretty much exactly what we've seen in the US this year, right? New York had a big peak in spring. All of the Sun Belt had sort of basically circulation over the summer without much interruption. And we'll start to see a resurgence now in the northern US in multiple places. In the UK in 2009, we've had a similar pattern with sort of an early summer wave interrupted by summer holidays and a second wave in fall, which this for SARS-CoV-2, this first peak was way earlier, was sort of in early spring, but sort of the resurgence after the summer holidays sort of looks pretty similar. This is Germany. Netherlands were pretty similar in 2009. Many places actually also skipped this first peak because the first peak was so far in summer already that there wasn't much of an early peak to be had, which only meant that the second peak sort of came very predictably in October. So the seasonal coronaviruses that I told you about before, so these relatives of SARS, they show pretty similar patterns to influenza if you look at their patterns over their circulation over the year. And that's sort of summarized here. So this is data from Stockholm, Sweden for these seasonal coronaviruses, which shows these sort of very, very regular oscillations with sort of peaks in December to February for the different subtypes. And we are in the yearly cycle now here. So you'd normally expect these guys to come back. But instead of these guys coming back, we'll probably see SARS-CoV-2 dominating the scene. My personal take is that the biggest driver of this is actually behavioral, in that we no longer sit out on the terrace in a cafe, but we go inside. We go in bars, we stick our heads together in restaurants. With our life sort of moving indoors from outdoors, the potential for transmission and the density at which people pack goes up. And this is what makes me and sort of many others believes that it will be much harder to control this virus through the winter. And I think we are seeing the first, basically, at least here in Switzerland, the weather changed quite dramatically 2 weeks ago, and we are seeing sort of quite the consequences of this, and it's not going to be easy in the coming months. So with this, I'd like to, skip all that. I'd like to sort of most and foremost acknowledge sort of my collaborator, Trevor Bedford, and his lab, with whom I've been working on NextStrain since 2014. There's lots of people involved in this, but in terms of SARS-CoV-2, there's a few people that I would like to call out, especially, which is sort of James Hadfield, Emma Hotcroft in my lab, Ivan Axamento, a programmer in my lab, Moira Suber, who has been helping with doing these analyses on a daily basis, and Tom Sibley in Trevor's lab, and also all the scientists and clinicians around the world that are helping with the response to this pandemic, and sort of sequencing all these genomes and graciously sharing them on the platform called GISAID. And with this, thank you for your attention, and happy to take some questions.
Yeah, I'm just gonna applaud. Well, thank you very, very much. It was super interesting. I can see that the questions are coming in through the chat. So Chase is gonna read them out, and then we're gonna have a few minutes for questions.
Okay, I'm reading one of the first questions here for you. Hi, Richard. Thank you for a great talk. Wonderful work. Although this may be slightly off topic, my questions are related to the effect of mutagenic antivirals on SARS-CoV-2 evolution. What do you think will be the effect of global scale implementation of mutagenic antivirals, such as vepipiravir, on the evolution of SARS-CoV-2? And to what extent do you think mutagenic antiviral drugs run the risk of stimulating SARS-CoV-2 evolution?
Yeah, no, that's an interesting question. So essentially the question is whether if we artificially increase the mutation rate of the virus, are we making it worse or better? And so these drugs are given in the hope that you screw up virus replication so much that the copies of the viral genome are mostly garbage as opposed to functioning virus. The immune system has similar strategies, specific enzymes that actually try to mess up with viral replication and puts in lots of errors. And we do seem to see signatures of this by the immune system. There's sort of an overrepresentation of particular types of mutations, which are quite suggestive of kind of immune system mediated mutagenesis. I don't think that we kind of help the virus along by mutating it more is a big concern, because the population of the virus is so big and the mutation rate is big enough that every relevant mutation is found thousands of times every day. And the dependence of the speed of evolution on the rate of mutation is not expected to be super strong in this limit. But it is certainly an interesting question to which I don't have a definitive answer, but my expectation would be that it's not a huge contributor.
All right, thanks, Richard. One more question from the chat here is, do you know what the reason may be for the geographic differences in epidemic curves? For instance, the double peak versus single peak signatures that we saw?
Well, sort of, the primary reason for different curves during the SARS-CoV-2 pandemic are different society and government responses in how stringently spread was contained. On top of that, I do think that double peak versus one single big peak, that potentially does have to do with just bigger sort of seasonal differences in different regions. In particular, in very hot climates like the southern US, the summer sort of drives people indoors because of air conditioning, which is not as strong a driver as sort of winter and heating, but potentially still enough to just keep sort of transmission in sort of a in a pretty high bracket throughout the summer, but never kind of as high as you would in sort of a northern temperate dense city.
All right. One more question here. Do you know why the influenza cases dropped in midwinter?
Well, like seasonal influenza has sort of the usual winter peaks. It sort of infects a certain fraction of the population and then sort of runs into existing immunity in the population, which sort of wears off slowly, both through virus evolution and sort of, turnover in the population and immune waning. And sort of in every winter, it sort of infects a certain sector, sort of 10-20% of the population, and then runs out of susceptible individuals, which put an end to these winter peaks. In the current context, we don't actually see any influenza because the transmission and that the infection control measures that are still in place seem to be stringent enough so that influenza can't get a foot in the door. We'll see whether that holds going forward throughout the winter.
Actually, I see that there are lots of more interesting questions coming up, but I'm sad to say that, well, I'm sad and happy to say that we're ending here. The questions will be posed to you, but we're going to the next speaker. So I want to thank Richard again. I was totally fascinated by the talk, so let's give you applause, at least us. And yeah,
my
thank
pleasure.
you. And then, okay, well, that's good to hear. Okay, so from now we go further with the next speaker. Next speaker is actually Kristina Ganzinger. She is at this moment a group leader at the Film Institute AMOF. She did her PhD in biophysical chemistry with Professor David Kleinerman on aggregation and segregation of proteins. And afterwards, she did a postdoc with Petra Schwille, I guess working towards synthetic cells. So I think we should just start because we're a little low on time. So without any ado, we're going to hear of Kristina about exploring information transfer in the immune system, reconstituting membrane-bound signaling process in vitro. So, well.
All right. Thank you very much for the introduction, Lidewij, and also thank you very much to everyone on the panel for having me today. It's a great pleasure to give first seminar from a Dutch scientist in this series this year. And I'm looking forward to many more of this kind in the months to come. So we are going to stick a bit with the immunology theme, although sadly it won't be much about COVID-19 this time. And when I talk to people what I work about, and I'm asked to summarize this in a single sentence, I usually say, I look at how cells in the immune system talk to each other. And this is kind of what the clay model that's depicted on the slide and was made by one of my former master students shows. Two immune cells interacting with each other. So why is this actually such a fascinating topic to study? Yes, okay, I think it's changed. So when we think about the immune system, we probably sometimes just envision it as this kind of shield that protects us from pathogens in the environment. But for example, I couldn't help the reference to SARS-CoV-2. We would really hope it protects us from being infected by this virus. But of course, mostly, even though we have a skin and we do have a protective layer that prevents us from being infected in the first place, most of the time our immune system really comes into action once we have been infected and the pathogen has entered our body. And as we just heard, pathogens are continuously evolving. So a real challenge of the immune system is that it has to dynamically adapt to be able to fight new pathogens at all times. And this is particularly difficult because at the same time, any error the immune system makes could potentially be deadly for the organism. This means that at the same time to being continuously adapted to the new molecules that maybe are presented on the surface of a virus, like the change in the spike protein, we still have to detect this with very high sensitivity and very high specificity. And overall, the response has to be measured. And this is also where we can see that things go very wrong with SARS-CoV-2. If you are suffering from COVID, this is exactly what essentially kills you in the end, is that your immune system overactivates. So this is also something that is very important. So if we think about how the immune system achieves this, basically, we could look at this from various different levels. And one would be that, of course, we have all those cells that you've probably all heard about in the last months more than before in our blood, white blood cells, immune cells that respond to pathogens and then essentially bring about changes and can even release substances to kill these invaders or infected cells. But all these kind of interactions between the cells have to be mediated on a molecular level. So in the end, it all boils down to signal processing on the molecular level of the individual immune cell, because each of these cells for themselves has to make the decision on whether or not to activate. And in the sum, this is really crucial that even a single cell takes this decision in the correct way, which also means that from a signal processing point, many of the processes in the immune system are really very interesting to study, because there's very complex mechanisms involved that really ensure that every input is correctly converted into an output from the cell. And this is what we are looking at. And of course, many other people do as well. And there's various ways of doing this. So for example, if you think about signaling, you think a lot about receptor-ligand interactions. So there are messenger molecules that bind receptor molecules on a cell surface. And then that's the first kind of signal transmission element of the chain. And you could study that by looking at structural interactions with structural biology or traditional kinetics using bulk biophysics measurement. And of course, we've learned a lot about these processes this way. And on the other side of the complexity scale, we could, instead of looking at individual protein-protein interactions, study these interactions in the context of a live cell, for example, where we can use methods like microscopy to really look at isolated processes, but they will be actually influenced, pushed around, and modified by all these individual processes going on in the signaling networks in the cell that essentially ensure that a certain output happens based on a certain input. But sometimes makes it very difficult to follow the path of this kind of signaling process. So what we are trying to do is to take the reductionist approach, but bring it to a more complex level by adding geometric context, going from a test tube to 2D or even 3D setups, and also making these systems more complex by having more components, really building up these signaling chains, kind of still in a test tube, but also preserving cellular geometries inspired by the recent advent of minimal cells and bottom-up synthetic biology. And of course, we are not the only ones who do this. And since I was also asked to give a bit of a flavor for where the field is going, I just thought I'd tell you why this reconstitution approach can be very powerful by showing some other people's work that I find very fascinating. So for example, several years ago, people have looked at the first steps in the intracellular transmission cascade in T-cell signaling, which is really at the heart of the system, the immune system or immune cells making a decision on whether to activate or not in response to a particular pathogen. So this is where the specificity aspect comes in. And they can really differentiate between, say, foreign and body-owned molecules. And here they were really showing that the spatial organization of these molecules in the membranes actually leads to phase transition effects that really affect the organization of signaling molecules and also affect signal processing in these structures. And that would have been impossible to see without the reconstitution approach. And it was also mammoth task, so they purified 13 different components. So that's already possible. You can also use this approach to gain more mechanistic detail about the signaling processes. For example, this is a different piece of work, different immune system process. This is about actually an innate immune system pathway, which is just a very general response to pathogenic bacteria. So this is just telling the body there is a pathogenic bacterium, yes or no, kind of a more binary thing. And here they could really show for the first time how the signal is passed on between various layers of molecules, which was completely debated before how this was happening. The last example that I want to bring, and don't try to understand this kind of complicated picture. Maybe I just wanted to show that that you can study quite complex systems with this. This is work from Jay Grove's lab in Berkeley, where they actually showed that a very important cancer target, which is this molecule RAS, gets activated by SOS, and this protein actually has an inbuilt kinetic proofreading mechanism, and they could really kind of show this for the first time using a reconstitution approach, which really tells us that at this stage already signaling is integrated much earlier than what was previously thought. So what are we interested in my lab? We, at the moment at least, focus on a signaling molecule called interleukin, which I would let's say very briefly describe as the accelerator of the immune system, or at least one of the accelerators. So there's a particular point when we have where we have to single out the correct immune cells for a response to the particular pathogen, because they can, for example, respond against a given virus or bacterium. And then this cell is singled out and multiplied because this is this adaptability of the immune system. And this multiplication signal is one of them, at least, is interleukin-2. And this is the molecule we are interested in. Because it turns out that interleukin-2 has a very closely related, let's say, cousin called interleukin-15. And both of these molecules, they are structurally quite similar. They bind also to almost the same receptor. So they share two subunits. The receptor for these two interleukin molecules, the beta and the gamma subunit, are shared. And so these two here. And they are also the ones that carry the signaling units of the receptor. There's a specific alpha chain that is different for interleukin-2 and 15, but that one is not involved in the signaling process. And the reason this is very interesting is that they show divergent function to the point that interleukin-2 introduces immune tolerance versus interleukin-15 introducing immune activation. So the question is, how can cells differentiate between these two molecules when the receptors are identical? And I don't have the data here, but it's very clear that structurally the complexes are really identical. So you can rule out any kind of conformational change-based mechanism. So the current hypothesis would be that one way to do this would be based on either different geometries, because interleukin-15 is mostly presented in a cell-to-cell contact. So this actually means that the diffusion is really different because you have two ligands restricted in the two molecules restricted in 2D versus a soluble three-dimensional ligand binding to a 2D surface. And of course, the kinetics could be different. And I talked before about kinetic proofreading. Not going to go into the details, but there are ways for a receptor signaling mechanism to really distinguish between ligands of different binding times. So this is the hypothesis that we want to test. And for that, we want to reconstitute the extracellular domains of these receptors in model lipid systems. And essentially, for that, we first have to make the proteins and, of course, also the lipid model systems, and then study them with total internal reflections fluorescent microscopy, where we can really follow the motion and the interactions and binding of all these molecules in real time. And then we can hopefully use this to answer question of what is the order of assembly of these complexes in the membranes? Is there any dominant species? And how are the assembly kinetics, and do they importantly differ for interleukin-15 and interleukin-2? And could this tell us how the cells can tell the two apart? So this project, I didn't introduce him yet, but this is all led by my PhD student, Kristina Ganzinger, you can see in the right-hand corner of the slides. And first, we had to express the proteins in insect cells because they are mammalian proteins and they can't be expressed in bacteria very easily. And we did this in collaboration with the NKI close to the VU, actually, in Amsterdam. And then we use an enzymatic method where we use sortase enzyme to attach a peptide tag with a fluorescent label to the protein. And then because it's a peptide tag, we also generate another site that we can use for purification. So you can really only purified labeled receptors. And then in the end, we can reconstitute them in membranes using an affinity between nickelated lipids and the his tag on the protein. And yeah, I mostly added this here because if people are interested in these kind of methods, then they should feel free to contact us. We have collected quite a bit of experience with this over the last 2 years. And then we also, at the same time, Christian built this amazing microscope, which has 3-color detection and a FRAP unit and a beam expander for having a very homogeneous field of view and loads of other really nice specs. So it's really like the kind of, I would say, the state of the art in turf microscopy. And if you are interested in getting some inspiration for your own setups or even building one at some point, we now have a very nice website hosted by GitHub where you can find all the information on the setup, from plans to component lists to everything, and of course contact information in case of questions. And with this, I want to show you some very first data that we took actually really recently, like 2 weeks ago. So this is a test experiment where we follow an antibody, where we follow a receptor in the membrane. We have a ligand present and an antibody that recognizes the ligand bound to the receptor. And yeah, and this is just a very first impression of what the data that we would generate would look like. So you can see this co-diffusion of the antibody with the receptor. And this is the way we would also generate information on the kinetics of binding and receptor assembly and so on, hopefully in the very near future for the system I was just presenting to you. And meanwhile, we also have some results, or we have some inkling why this might be interesting. And that comes from theory and from a really nice collaboration with Peter Reinthen-Bolde at Armolf, where we have already simulated this system using the Green's function reaction dynamics approach that they've developed over the years in his group. And what you can see on the right-hand side in the graph, which is some initial results that a previous master student, Tom Clement, got, where he varied computationally the ratio of alpha to beta chains. And then also there was, we had some, all the kinetic rates are estimated from 3D kinetic measurements that were available in the literature. And we have some interleukin-2 present, and now we can see which kind of species are forming, depending on how much alpha chain compared to beta chain exists. And what I need to tell you maybe before is that historically people have thought that this interaction here is really the capturing interaction because it has a really high affinity. So the alpha chain has a very high affinity for the interleukin-2. So people were convinced that this was essentially how interleukin-2 gets bound to the cell, and then essentially the alpha chain transfers it to the beta and the gamma chain. But there are also affinities of interleukin-2, different combinations of these molecules, and also independently to beta gamma, independently of alpha. And it turns out if you actually simulate a system taking the 2D geometry into account, that there's almost no interleukin-2 bound to the alpha chain. So this would be this brown population here, which you can almost not see. And on the contrary, actually, a lot of interleukin-2 is bound to the to the beta gamma unit alone. And the reason that, so this is a really interesting result, and we are also very curious to see if we can find this with our single molecule experiments, of course. And because actually people have really targeted this alpha chain for therapeutics, and it hasn't been so successful. So this might be one of the explanations why this is maybe not the best strategy. And also another reason why looking into kinetics with that level of detail could even be pharmacologically interesting in the long run. Now, one big problem that you have when you try to find out kinetic or measure kinetics with single molecule methods is that your fluorophores will eventually bleach. So you get a certain window of time and a certain time resolution that you can get that is determined by this bleaching, because you cannot have signals for very long. So like long time scales are basically inaccessible. So over the last years, in collaboration with my postdoc lab, we actually set to work on a method to essentially improve the temporal resolution of single particle tracking by repurposing a method called DNA paint that has been used for super resolution microscopy before. And basically the idea is you label your molecule of interest with a long DNA molecule, and that has several binding sites for smaller imager strands with a fluorophore. And now you turn or tune the binding rates and the K on and K off of these imager strands such that on average, when a molecule bleaches, it's also replaced again by a free imager from solution. And of course, because this is, if you use turf, you don't really bleach the molecules in solution very much at all. So this will be almost always a molecule that is fluorescent. So in theory, you could have infinite trajectories that way. And it actually works very nicely, as you can see here. So this is just using a single dye label, which is what we also have been doing before. And you can really see it bleaches already after 20 seconds or so, which is quite typical and not a particularly bad result, I should add. Whereas if we use this new labeling method, yeah, we really get signals that last for minutes, up to half an hour or even 2 hours was the most extreme case we witnessed. And if you are interested in this kind of method, then I direct you to our preprint on bioRxiv for more information and more technical details. And of course, you can also contact me personally. Now, to finish, I just want to give some brief flavor of how we want to even increase complexity in the future by going from 2D to 3D using giant unilamellar vesicles instead of supported lipid bilayers as a kind of lipid model system. Because of course, if you think about signaling processes and signal integration in cells, we think about a confined environment, and this will also change the boundary conditions. So ultimately, it will be very interesting to reconstitute these reactions inside a lipid vesicle of the size of a cell. That's of course very much in line also with what the basic consortium here in the Netherlands try to build a synthetic cell, which also ideally at least should be able to process signals. And in a project led by my PhD student, Lori, we made some steps towards improving the reliability of a method to make these vesicles, because that's really at the moment a technical problem in the field that's really dominant, that we just really can't make these model systems very well at all. And the method works as such that you spin a disk, first with some aqueous solution, then you add some lipid in oil solution. So you have a two-phase system, and you add a capillary. And whatever you want to encapsulate, you essentially shoot with the capillary into this oil phase, so little droplets start to form. And first they essentially acquire like a monolayer of lipids from being a droplet in a lipid and oil phase. And as they pass through the interface, they actually obtain a second layer and turn into a lipid vesicle. And this method has been known for like almost 10 years already, but has not really been taken up by the field so much because it wasn't working so reliably in many people's hands. This is by how the system looks in real life. This is the setup we built. Again, we share all the plans via the GitHub website. And so when we actually looked into optimizing it, first we had to do quite a few things to make it work, but I'm going to skip those details, technical details. We also looked at what now we can really have thousands of vesicles in a single run. We can really look at things on whether the rotation speed matters or the size of the capillary. And we actually found that it's quite robust with respect to these conditions. So you can see that all these distributions look very similar. And in fact, the variation from run to run is bigger than, or at least as big as the variation between these conditions. But on the other side, other factors that may not be so obvious at first to vary do actually influence the system very much. So for example, the humidity in a room that we do the experiment in, which you normally don't really control, right? You control this maybe in a clean room, but you don't control this in a lab. Maybe you control temperature if you're lucky. But if you don't control the humidity, then it turns out that in the wet and humid Dutch summers, your experiments just stop working because your humidity in the room goes over 60%. As you can see from these pictures, the yield just drastically drops as humidity increases. So we actually now use a dehumidifier to stabilize our experimental conditions. And we also found that this lipid and oil solution is actually really unstable because you use a mixture of solvents that some of them are very volatile, and that essentially generates a very unstable mixture even on the time scale of the experiment that you're running, which I also don't think many people, I don't think that many people are aware of this. And it is something that also means that you have to work very quickly and really make everything fresh for every single experiment. And there's still some remaining unknowns that influence yield, but by and large, if you keep these things in mind, the protocol works really quite well, at least in our hands and also in the hands of people in other labs in the Netherlands, because we have collaborated with Gijsje Kunderwing's group now at the TU Delft for this, to also show that the vesicles we produce are unilamellar and have kind of oil-free membranes because hemolysin can insert into them. So it's a kind of indirect evidence for the oil-freeness. And we can reconstitute an actin cortex in these vesicles. We also collaborated with Kees Stekker's group, also at the TU Delft. They have reconstituted synthetic cortex based on DNA nanostars that are recruited to the membrane in the presence of cholesterol in these vesicles. And we can also encapsulate SUVs to generate a multi-compartmentalized system. So it has at least worked robustly across 2 places and 3 labs over the last year. So I'm quite hopeful for this being quite a useful method also for BASIC in the future. So finally, I just want to say a few words about the challenges ahead to kind of come full circle to the beginning on why we would want to do this. So if we really think that having a minimal like signaling cell system or like immune cell maybe is interesting, what are the challenges? Well, I think many are on the biochemical side, like to do with having, if you want to increase complexity and have all these different protein components, and for a signaling pathway, ideally you need really like, let's say between 8 and 10 to have a sufficiently complex system, then they need to be all expressed, purified, they need to work in the same buffer, they need to be still functional, etc. So this is a big challenge. Also making lipid systems that are compatible with the conditions is hard. But there are also some challenges on the data analysis. This is just an image showing tracking and how this can obviously be a problem, but generally working with microscopy, we think a lot about image recognition or extracting features from imaging, which has really its own challenges. And there I also think a lot of improvements can maybe come from more automated methods or even using machine learning in the future that could be very useful for the field. And finally, of course, in the end, you would like to bridge back to the more multi-system level where you have not just one single cell system, but you have actually cells talking to each other, meaning minimal cells maybe talking to each other. And there have been some first really nice attempts published this year by the Van Heest lab from the TU Eindhoven, but in general, anything that has been done there is for very simple systems. So I think this will bring fantastic insights, but will also be a challenge to achieve these kind of things for more complicated cellular signaling pathways. And yeah, with that, I really want to thank my group for being an amazing group to work with and making everything very enjoyable, but also our collaborators, both at AMOF and at TU Delft, and you for your attention.
Yeah, thank you very much. It was a super fascinating talk. I see that there are no questions in the chat yet, so maybe Jace, do you want to start with a question or shall I ask?
Yeah, I'll maybe start with a quick question. So first of all, Kristina, thanks for taking on the challenge to tell us about where the field is going and the bigger challenges ahead. I was kind of interested in these interleucins that you were talking about. And I was wondering about, to get a bit more context there. So you looked at these 2 specific ones, interleucin 2 and 15, which had such a different function. And you you were speculating whether this differentiation happened based on spatial context, right? And so I was wondering how common this is. I mean, like if you look at different classes of such receptors, I don't know how many there are, but how common is it that you have such similar receptors with such divergent functions that you really need additional, for instance, spatial context as an additional differentiation mechanism?
So I think it is more common than people think, but all the canonical examples are, of course, different. So this is the textbook picture that we have from signaling. Is usually the ligand introducing a change, or there being a single ligand receptor kind of unique interaction, or like a dimerization or something like this. However, particularly in the immune system, there seem to be quite a few cases where this may not be sufficient, because the body essentially recycles already existing molecules for a new task, repurposes them or uses them double. And also there are receptors like the T-cell receptor, which essentially have to react so specifically and sensitively that they also need additional mechanisms rather than just like a binding yes or no, to distinguish between a quality of binding events. And then again, you have to have an additional mechanism that ensures that you can differentiate between certain types of ligand binding events and others, where also kinetics and also actually spatial context can come into play. And that's also what people start to think more and more.
All right, thanks.
Actually, so because you use DNA paint, right? So you can trace your proteins, your membrane proteins longer, but it's also a sort of larger complex. So I'm wondering whether you are actually able to detect the sort of diffusional properties of your protein. Then do they change in a way that is observable, or is that not something to worry about?
No, so so we didn't see a change, and actually we did test that for the test sample that we used in this preprint, which is just simply a molecule actually, or like a DNA origami on a lipid bilayer. But there we have quite a strong attachment to the membrane, which dominates in a 2D motion case. It really dominates the diffusional properties. So the 3D parts sticking out in solution, it doesn't really have a big influence on the motion. It's maybe slightly different if you have a single, let's say, amphipathic helix connection, for example. Then you may see a slight difference, but it still would be expected to not be very, very large, the effect of the tag.
So do you think your DNA doesn't have any interaction with the membrane, basically?
No, this is definitely not what we saw. Yeah, no, we didn't see that. Yeah.
Thanks.
One more question from the audience. Sorry, Tom, go ahead.
DNA tags with each other, do they? I guess you have to worry about that too, right? That they might hybridize and so on. But maybe the design space of the sequence is big enough that you don't really.
Yeah, indeed, this is exactly what you have to take into account when designing them, and also the buffer conditions matter. So you don't have full flexibility over your buffer, because the ionic strength of your buffer also matters. It has to be kind of enough to suppress unspecific interaction, and but yet low enough to allow for specific interaction. So there's some tuning there as well. Yeah.
But I guess usually you don't have more than a couple of colors, right, at a time. So as long as
maybe
don't,
at most, and
So I guess given the sequence space, you're pretty good in the design.
Yes.
Space.
So we are currently looking at 2 different designs which seem to be orthogonal. We haven't yet tried 3, but I also think that there should be space for that. Yeah.
Nice.
Yeah.
One question here from the audience. Thank you for the nice presentation. I think in the method you have shown for making GUV, you will have an oil layer between the lipid bilayer. Since it is impossible to get rid of the oil from the lipid tail, could you detect it?
Yes, so this is a question that keeps coming up actually, and so there's 2 answers to this. Answer number 1 is that the original publication says that that they are really sure that there is no oil layer, and they have made certain measurements to show this. The other thing is we could certainly try to, or we will actually, in fact, do some mechanical measurements on these GUVs to show how similar or how different the properties are compared to electroformed GUVs, and then we should really have a very good idea of how much there's still an oil layer. But the data I already showed on the hemolysin suggests that there's certainly not a lot, because the protein is extremely sensitive to the amount of oil in the membrane, so it cannot insert functionally unless the lipid bilayer is pretty much a proper lipid bilayer. So there may be still residual molecules for sure that we can't rule out, but it seems unlikely that it's a lot at this point.
All right, thanks.
So I think with that, we come to the end of these 2 talks. I want to again take the opportunity to thank Kristina and Richard for being so brave to give the first 2 talks in a seminar. I tremendously enjoyed it. I think I get way too little science talks. So thanks again very much. Maybe applause. Let me say 2 more things. So the first thing is to tell you that this is the first, but definitely not the last of the series. So the next one is going to be on November 10th. And November 10th, we're going to have a talk by Cliff Brangwyn and with one of Nama Brenner. So I think they're also really awesome speakers. So I recommend you to come as well. The second part is that we'll now switch from the seminars to tutorial. So this means that, I mean, anyone is basically happy to stay, but there's people who registered for this tutorial and they can now join as a panel member. And this means that they can directly ask questions to Richard during his tutorial. So I see people coming in. So let me thank all of you again for coming, those here in the Zoom session and those on YouTube. And we'll now continue with the tutorial from Richard as soon as people have arrived. And the tutorial will basically be about, quickly look this up, modeling and predicting RNA virus evolution. So yeah, Richard, floor is yours.
Yes, thanks again, I'm still here. Let me share my screen again so we can we'll get going.
Oh,
yeah,
Richard, while you're setting up, I just wanted to say I saw your next strain work early on. This is Greg Stevens, as one of the panelists. I saw your work early on because my family lives in Seattle, so I was watching the epidemic kind of unfold in Seattle, and what I learned from from Twitter and blog posts from Next Strain and so on was really impressive. So I really appreciate the work. Thank you for doing it.
Yeah, thanks so much for saying this. This is always very gratifying to hear these these kind of testaments on how it's making a difference. Thanks a lot. Yeah, but in the second part of the talk in this tutorial, I was going to take a bit of a step back and sort of ask more basic questions about RNA virus evolution. It will also have some elements of tracking, but largely I'm sort of trying to get a little bit into the theoretical models that one uses to understand virus evolution and see how sort of fitness relates with growth, antigenic evolution, success, and so forth. And this will be mostly focused on influenza, because influenza is sort of the virus that we've been studying for many years, where there is sort of a huge public health interest in sort of developing vaccines and things like that, which is of course now all sort of dwarfed at some scale by SARS-CoV-2. But in the long run, influenza will probably be something that gets to be center stage again, and it's a good model to understand more general features of RNA virus evolution. So, some of you will know this. This is a basic summary of influenza pandemics of the last 102 years. And, all of you are going to be aware that there was this, the devastating so-called Spanish influenza. Doesn't really have much to do with Spain at all, but that's how we end up calling it, where sort of probably from an avian source, an influenza virus, jumped into humans and caused in multiple waves a deadly pandemic that, in contrast to SARS-CoV-2 now, sort of mostly, or at least sort of strongly affected people in their 20s and 30s, which was sort of quite unusual, because these are usually, people that sort of don't really suffer too much from that kind of respiratory disease. So after multiple pandemic waves, the virus then settled into sort of a seasonal pattern that I showed you earlier, where it sort of comes back every winter, sort of causes an outbreak, which is mostly affecting the kids and the elderly. And this sort of went on until the late 50s, when a new strain of influenza appeared, which then was called H2N2. H and N sort of, they codify the two different surface protein of the virus. So this was just version 2 of both of these, which were sort of classified by serological sort of immunological assays. And this H2N2 is sort of displaced H1N1. And then sort of continued to circulate for a few years before being displaced by yet another zoonosis, another pandemic. So each of these big circles here are pandemics in a way, and then in 68, the so-called Hong Kong flu sort of displaced the Asian flu H2N2, which is a variant of flu that is still circulating today. And in fact, it is a variant that causes the more severe influenza waves in every other year or so. The H1N1s, these descendants of the Spanish flu, they sort of somewhat mysteriously came back in the late 70s. And they came back in a way that is completely unchanged from back here, which is nothing that sort of RNA viruses typically do, other than if they're kept in the freezer. So there must have been some sort of release of this virus in some way or another, which the details of which are not, at least not publicly disclosed. But yeah, this virus continued to circulate until 2009, when swine flu came about. Swine flu is sort of a colloquial name for an H1N1 variant, which actually has at least some segments of which have a common ancestor with the Spanish flu. That's one of the reasons why people were so scared about the 2009 pandemic influenza. But interaction between these two strains was sufficient that this 2009 strain of influenza drove the original Spanish flu strain of influenza extinct. And currently we have these two influenza A lineages in circulation, which cause these seasonal waves, which, this is a graph that you've already seen, which sort of cause these winter epidemics. And here in green is sort of the H3N2, so that's the one that's been circulating since 68. And that's the one that's dominating most years. The original H1 lineage that descended from the Spanish flu is in blue, which has been sort of dominating a few years, but typically is subdominant. And this has been replaced by the 2009 pandemic influenza lineage in yellow, which caused these sort of big anomalous year here. So that is a pandemic year, which resulted in a very early and severe, or at least high case number flu season in 2009, 2010, and then sort of a bit of a low for a few years before the H1N1 also settled into the seasonal pattern, sort of oscillating with H3N2, but H3N2 tends to be more prevalent. In red, we have influenza B lineages, which are quite distinct viruses, but they also tend to cause outbreaks a little later in the year usually than the influenza A.
Could I just ask a question about the previous slide?
Sure.
The one before this. I was just wondering when the original H1N1 returned in the 80s, did it cause a lot of deaths like the first time? I mean, or was there so much background immunity that it was fine, or was there another reason why?
It basically just slipped back into the seasonal pattern. So there might have been a bit of an anomalous season or two because of some fraction of the population being completely susceptible. But yeah, no, it didn't cause anything nearly as bad as before.
But is it known that's because of background immunity, or?
That is, certainly the most vulnerable people, they would have been exposed to this, very similar viruses 20 years prior. And so the very, the severe nature of the Spanish flu itself is not, terribly well understood, but probably has to do something to do with sort of an unfortunate immune reaction towards the similar antigens that people picked up in the 1890s or something. So it's still a little mysterious why this has this anomalous mortality curve, and it's not expected to repeat itself simply because of different immune history in the 1970s.
Okay, thanks.
Yeah, so I'll be mostly talking about influenza H3N2 because this is a strain that tends to adapt and change most rapidly, and where we have the most continuous and long time series available. So what you see here is a phylogenetic tree of about 35 years worth of influenza H3 and 2 evolution, with the very old strains here at the bottom, bottom left, and the more recent ones. This slide is not super up to date, but you get the idea. In the top right, you get sort of the more recent strains. And as we move from left to right, the virus accumulates mutations, and it actually accumulates mutations sort of quite rapidly, in that these viruses down here are about sort of 10-20% diverged in terms of nucleotide identity from those more recent ones. These nucleotide mutations, they tend to change amino acid in the head domain of the hemagglutinin protein, which is the equivalent of the spike protein for influenza. So it's an envelope protein that sticks out of the virus and mediates entry into cells. And it's also the primary target of human antibodies, which tend to target this head domain, which as a result sort of changes quite often a bit. It has sort of a whole bunch of sites which seem to be very flexible in evolutionary terms, which can be used as sort of decoy sites where the virus is sort of changing its properties quite often without really harming function. And yes, so you see, especially here in this crown domain, that the virus evolves very rapidly, and the result of this is escape of existing human immunity. And the side effect is that the vaccines that we get, the flu shots, need to be updated very frequently. And in fact, these gray crosses that you see on this tree, they are representatives that were chosen by the WHO as strains that go into the flu shot over the years. And in many ways, sort of the vaccine producers and the WHO, they play catch up with the virus, that are always sort of trying to provide a vaccine that's close enough to the circulating viruses so that it works. To give you some sense of the dynamics of this, I here plotted the amino acid the frequencies of different amino acids at one particular position. That's position 189, sitting somewhere up here on the protein, which changed identity about 4 times, well, exactly 4 times over the past 35 years. Starting out with the lysine, going to arginine, serine, and so forth. And what you see is that each of these takeovers took about a year or so, 2 years, and then sort of the complete flu population was replaced by a new population that had these different properties. Sometimes these mutations happen in parallel on multiple branches, so that the depth of the tree is actually a little more. And this is to be fair, a little bit of an extreme example, but there's sort of multiple other positions, dozens of other positions that also change over this time frame.
Richard, just a quick question about these trees. To what extent do we ever need to worry about the extent to which this is sufficiently sampled and sequenced such that we have a complete picture of this genetic tree?
Well, that depends a little bit on what the question is that you're asking. So, influenza, at least, pre-SARS-CoV-2, tends to be fairly well mixed, in that sort of pretty much every continent has a representation of every major variant. And then sort of, the large clades you'll all pick up, even with a fairly small sample. Of course, sort of, rare variants, you need a deeper sample. That's kind of a tautology. Generally, the deeper you sample, there is sort of diminishing returns, in that you tend to find more of the same and not so many new things. That depends a little bit on the structure of the tree, but at least at present, we have a pretty good sampling with sort of hundreds to thousands, certainly thousands of sequences a year, sometimes hundreds a week, such that we have a pretty good understanding of what's going on. There are sort of geographic white spots, sort of like, South Asia, for example, is something that's a little bit difficult to assess, despite the fact that, a good chunk of the world's population lives there. But overall, we have a pretty good representation.
I have also a question. So I guess for this tree building, you assume that there is no recombination somehow for the virus, right?
Yes, and that is true for influenza segments. So there's no convincing evidence that individual influenza segments would recombine. Right. That said, influenza has multiple segments, and different segments resort and have different trees.
Right. And I was just curious, like, for example, for the COVID, is it already clear that they cannot somehow recombine?
No, so coronaviruses do recombine.
I see.
And it is quite possible that there is already signal for recombination in these wealth of sequences that we have. Currently, however, it doesn't seem to mess up things in any sort of meaningful way. I don't think anybody has made a very systematic investigation of this, which, you'd only expect this to have a dramatic effect if you have enough diversity and high infection force that you get sort of co-infections of individuals with pretty distinct strains. And I do expect this to become important in the near future, but currently this doesn't seem to make a huge difference yet.
So what's going to happen in the near future?
Well, we'll have to screen these genomes for being recombinants. We'll have to maybe sort of do separate phylogenetic analysis of different parts of the genome.
Yeah, I understand. Yeah, I understand. But the reason you say near future, that's because the infection rate goes up. So that you're getting two different.
Yeah, well, both diversity increases, right? And the more diverse the population is, the more impactful you expect the recombination event to be.
Right.
And with sort of the higher the prevalence, the higher the chance that it actually makes a difference, that it happens.
Right, and then it could affect your analysis. Okay.
Then it would make the analysis a little wonky, and we need to keep an eye on that.
Yeah. Okay. Thanks.
I also sort of have a quite elementary question about the graph that's currently shown, also related to the previous slide, but I guess this is very striking what you're showing here. Every time there's a new mutation, it takes over quite rapidly, right? I mean, you said it's about a year, but I guess, what's very striking is it looks like, not only is the new one, good at spreading, but it's also, spreading at the detriment of the previous strain, right? So, and maybe that's just obvious, but I mean, I guess it suggests that there's a finite niche that they kind of take over, right?
Yes, no, that is the correct interpretation. And the next slide, I still have a bit of a counterexample to that that dynamics. So we know that these viruses interact antigenically, right? Sort of in order, if one person is infected, it will have antibodies that cover some sort of 10, 15 years worth of evolution. And in any given year, so only a fraction of the population gets infected. Yeah. And in some ways, the range of antigenic cross-reactivity is bigger than the diversity of strains currently circulating. So in some way, that means that all the circulating viruses are competing for the same resource, namely the people that are that haven't been infected a long time and don't have immunity to current circulating variants, with sort of incremental improvements by adding additional mutations.
Yes, that's quite interesting, right? Because I guess they're triggering, an immune reaction that will kill the.
Yeah, and at some level, there is sort of a weaker interaction that actually goes a lot further still, which is probably mediated by T cells, which leads to interactions on between much more distinct strains, which, for example, is hypothesized to be due to the kind of the extinction of the original Spanish flu descendants by the swine flu, which is it is quite it's 100 something years away from that, right? But there's still sort of residual cross immunity, probably mediated by T cells, that meant this interaction is sufficient to drive the other one out.
That's remarkable.
But since we are on the topic, so this is a tree for H3N2 that I just showed you. If you look into influenza Bs, you find a somewhat different lineage. Of course, it's just one example because there's only so many flus that go around that we sequence. But influenza Bs, they sort of split at some point in the 70s into two lineages, which have since been circulating more or less independently of each other. There is some sort of oscillation between them, but here sort of exactly this happened what we just discussed, that one lineage seems to have managed to get away from the other one sort of far enough in absence of competition that they then sort of circulated independently, and somehow this niche overlap is reduced, and they're kind of competing for separate fractions of the population in a way.
Is it because there's separate populations, or is it because they've diversified sufficiently?
Yeah, so it's, this, we're essentially talking about things that happened in the 80s, right? Sort of what sort of allowed these two lineages to get away from each other far enough that they essentially immunologically decoupled.
I see.
And the sampling in this period is pretty sparse. This could have been due to also, if they sort of take foothold in different geographic regions and there's not so much traffic, that of exchange between these regions, that certainly helps developing these different scenarios. One thing that is quite striking at present is that one of these lineages has a very different age distribution from the other. So it looks like one of them is mainly going after the kids and the other one is mainly going after the elderly. And so that might be sort of not only a change in sort of antigenic properties that they decouple, but maybe also a change in sort of quote unquote strategy in sort of what their sort of target demographic is.
I see. Yeah. So there's.
Anyway, sort of, yeah, I already talked about this. So we have a pretty decent representation of the globe in terms of sequences, which used to be completely unique until the beginning of this year. Now with SARS-CoV-2, we have a similarly and even more impressive collection of sequences. This is sort of all federated by a global network of influenza research labs and sort of a database called GISAID, which sort of facilitates this sort of rapid sharing of sequence data in a very timely sort of pre-publication, often within 2-3 weeks of sampling. And yeah, in winter months in the Northern Hemisphere, we typically have hundreds of sequences per week. And sort of what then sort of, with these amount of sequences, what became challenging is sort of how do you actually sort of analyze these in a sort of a timely manner and disseminate the results, because what these different labs sort of they used to do when they only had a few sequences is build a tree, sort of print it out on paper and show it around, which really sort of wasn't that feasible anymore with that amount of sequence throughput. And that's where sort of the Next Flu project, which is the precursor of NextStrain, sort of came about. And I'll just have a little screenshot here. You already seen some of that. This is a now somewhat dated analysis, but the basic points are still correct. Where we have a pretty decent representation of the globe. We try to sample this as representatively as possible, as we can't analyze everything at once. There's some geographic stratification here. For example, that is much more Americas than these groups over here, but generally you see that everything is everywhere in some way or another. I could sort of go through the features of the site, but I think in the interest of time, I'll just sort of move on and invite you to play with the different features. You can, for example, color the tree by amino acids at every position in the genome. You can look at the age of the people in the different clusters. You can zoom into the different clusters and things like that. Now, what I want to get into is kind of beyond tracking. So what I've showed you so far is essentially sort of an infrastructure initiative where we sort of set up tools and pipelines so that we can ingest the data as it comes in and display the results, disseminate it on the web in interactive fashion to those that are interested and have to make these decisions. But we wanted to go beyond that, so right, sort of asking the question: is there something that we can do beyond taking snapshots? Is there some sort of some way or another that we can extract sort of dynamical information from those data and use that to predict, to sort of extrapolate into the future? And we were actually asking sort of an even more difficult question. We were sort of asking, if we only had a snapshot of a bunch of sequences, all from the same time, what can we say about how these things are developing? And to get there, I'll have to make sort of a slight detour into sort of models of population genetics. And sort of our basic framework here is going to be the following: that we consider a population, again, sort of black lines are genomes, the colored dots are mutations. We consider a population that has a fair, substantial number of these mutations, all of which contribute to fitness in some in some sort of well-behaved manner, meaning there is sort of a large number of mutations that have similar sizes, similar effects on fitness, in which case we get a fitness distribution in the population which is not too jumpy. It's somewhat bell-shaped, with sort of high fitness individuals by definition sort of growing in frequency, replacing those with low fitness and so forth. Right, that is sort of the the prototype of these sort of standards of traveling wave fitness models of rapidly adapting populations. And there's a whole bunch of theory that you can do on this, and this has entertained us, so Michael Desai, sort of Kessler Levine, Igor Ruzin, and a bunch of other people over the last 2 decades or so. It's been a very fruitful development of theory, where sort of a couple of general lessons have been learned is that the speed of adaptation, once you have a large population, it's actually fairly independent of the population size, only depends slowly on or logarithmically on that. Similarly, on the mutation rate, it depends fairly slowly. And that is because most of the adaptation is sort of limited by competition between multiple variants and the degree to which the environment selects them, rather than sort of by a limitation of finding the mutation. So it's not so much mutation or discovery limited, but sort of competition and sort of bandwidth limited, which results in these sort of logarithmic dependencies. And another thing that we've learned is that there's sort of many different sort of variants of these models with sort of steps, fixed size mutations, exponentially distributed mutation distribution, things like that. They all end up sort of having a number of universal, almost universal properties, which distinguish the genealogical properties of the diversity properties of this population quite systematically from common neutral population genetics models. And this is sort of summarized a little bit here. On the left, you have a screenshot of a tree that is generated by neutral simulation. And here, I apologize, so the present is at the bottom, the past is on the top. That is the root of the tree, which obviously has to be at the top rather than at the bottom. But evolutionary biologists sort of plot trees in all orientations, only never with the root at the bottom. The root sideways, root at the top, that's perfectly fine, but root at the bottom, you essentially never see, for whatever reason. But yeah, this is kind of a prototypical neutral tree where these lineages at the present sort of come together into common ancestors very, very quickly. And then there's a few sort of long lineages that go back to the root. And here on the right, I sort of contrast this with a tree that's sampled from a rapidly adapting population, where you have many mutations that compete, that sort of makes these individuals fitter, and you see sort of a bunch of quite distinct behavior. First, for a long time, there's nothing much happening. These lineages, they go forward, they go back into time without merging, and merging only happens once you're fairly deep in the tree. And then you see sort of bursts of coalescence, which happens sort of quite deep up in the tree, coupled to a branching pattern which sort of sends pretty much all individuals down one branch and a minority of individuals down the other branch. So here you have 1 and sort of 29 going the other way. And this is sort of a nested fashion. You have this sort of tree, which sort of a bit like a Russian doll kind of thing, where in the way this is displayed, the right branch already only contains few individuals and the left branch contains most individuals. And this, in fact, has sort of a fairly intuitive explanation that can be put into complicated mathematical models, but I'll just sort of try to get you the gist of it. So, when you sample individuals at the present, they'll mostly be somewhat ordinary, middle of the road, bulk of the fitness distribution individuals, right? And there's many individuals in the center of the fitness distribution. This is a place where most individuals are. And since there's so many, the chance that any two lineages meet and merge is pretty low. So what you observe at the tips is sort of an absence of coalescence until something happens to where these lineages are. If you trace them back in time, you'll notice that the fitness of the lineage relative to everybody else in the population is constantly increasing as you go back in time. And this is an effect of conditioning on having offspring, right? So all of you have parents, but only a fraction of you have kids, right? So by conditioning on being a parent, you sort of move yourself probabilistically into the high fitness end of the distribution. And if there's many sort of small effect mutations, this is sort of a fairly deterministic backwards drift, which sort of crams your lineages into the high fitness tail of the distribution. And only once you're there, where the number of individuals that you're competing with is smaller, that's when sort of coalescence sort of starts to set in. And then once all these lineages are sort of crammed into this high fitness tail, this is then when the actual competition dynamics happens, when sort of one individual sort of happens to pick up a few mutation and increase in fitness. It ends up being ahead of the pack, can expand exponentially because it's growing faster, and take over a substantial fraction of the population. And this is what generates these sort of bursts, these merger events. So these merger events correspond to transiently high fitness individuals. And you can sort of go through some calculations and show that if you have these exponentially expanding clones that sort of descend from kind of lucky mutation, jump ahead everybody else for a little while, you get sort of effective offspring distributions over a short period that are power law tailed. And these power laws, they generate the statistical features of this tree, including this very skewed branching, where sort of the rare event can sort of absorb a large fraction of your population going forward. Yeah, and this was sort of one of the core realizations that we had there, where that these bursts of the tree, but they correspond to at least transiently high fitness individuals. And then we are sort of trying to turn this around by looking at the tree, and from the tree alone, tell us which parts of the tree are likely to be more fit than others. And we explored this first in simulations, but let me introduce the setups that.
Sorry, just one question.
Yeah.
So how does the interaction with the immunity of the population come into this fitness picture? Because if I think about like a burst of some virus, say, that kind of implies that, well, it's spreading quickly. So also a lot of people will build up immunity, which should, in a sense, then decrease its fitness at a later time period, right? So how does that come into the picture here?
Yeah, so one is here sort of looking at sort of an endemic situation where some fraction of the population, which is comparatively similar every year, sort of gets infected, and immunity in the population as a whole is sort of keeping up with the virus population. That the virus sort of picks up a new mutation, which makes some sector of the population susceptible again, and then it sort of infects that part. Then there's a new mutation and sort of a new sector of the population. So this is not meant as a model for kind of a pandemic outbreak scenario, but more as a model for an endemic virus that's continuously circulating.
Okay, thanks.
Yeah, anyway, sort of, but currently we are really only just talking about sort of a more abstract model of population, and we'll see to what extent we can use this in the end to apply to flu. And it turns out that we to some extent can, but maybe not as much as we had hoped. So, but what we are sort of trying to do is sort of assume we have a slice of a population at some time point, all individuals sampled at the exact same time. And we'd like to find the representative at the present time that is sort of closest to some future population. And the two are connected through a common phylogenetic tree. And there is sort of one lineage, which is sort of the backbone of the tree, kind of the root of the common ancestor of all of the future, which tends to run always through the high fitness tail, right? Sort of whoever the ancestor of everybody in the future is with very, very high likelihood always a very fit individual. Now, obviously, in this case, it would be this individual here, which is sort of most closely connected to the future population, but nobody tells us their ordering on this fitness landscape. This is what we are trying to infer from the data, right? And sort of we devised sort of an algorithm for this that sort of tries to, yeah, in that we write down a probability distribution for the relative fitness of every node in the tree, every terminal node in the tree, but also the internal nodes. We try to write down a probability distribution to observe a particular configuration of fitness on the tree, conditional on the tree topology and the branch length. And you can do that by sort of attaching an object, a four-dimensional object, to every branch in the tree, which quantifies how likely it is to have a certain fitness at the parental end of the branch and a certain fitness at the child branch, and observe offspring at present and no other branches and no other sort of events that happen between along the branch. So there's a bit of a sort of complicated object that you can approximate by sort of a branching process. You're essentially asking if this thing is dividing with a certain rate, how likely is it that I observe anything in the future, but no other descendants along this line. So it's a bit of a complicated calculation, but you can attach such an object with every branch of that tree, and then marginalize this effectively because it's a tree, and you can do the recursive transfer matrix type operations. And in this case, this would sort of single out as highest fitness this little burst here, where this lineage sort of rapidly spread into multiple lineages, which left 5 offspring a short time later. So it turns out that there is also, to this kind of bursty picture that I pulled up, there is a fairly simple heuristic, which in some real world cases actually works better than the complicated formula, where you in a very computationally efficient way, you can assign sort of a score to every part of the tree by calculating a particular weighted integral over the tree, which we call the local branching index, and which is essentially just a measure of the local bushiness, of the local burstiness of the tree. And it's sort of illustrated here how this works. So you have the usual tree here with a root at some point in the past. If we just unroot this tree, meaning we remove the special feature of this node, it's the same tree that you see over here. We then, for every point, sort of just ask how much tree volume, how many lines, what's the length of all the lines in some radius around it, and instead of choosing a particular radius, we do sort of an exponential integral over the tree, which is again very easy to compute, and you have this sort of score which measures local branchiness, which is a proxy of fitness, which one can deduce in some asymptotic limits from these formula here. But just bear with me. This is sort of a useful simple heuristic that picks up most of the features of the fitness prediction. So we tested this in simulations, and that's obviously sort of an easy thing to do. You simulate a bunch of sequences, simulate evolution, you sample a bunch of sequences, you reconstruct the trees, knowing only the ACGT sequence that your simulation program spit out. You then can apply this algorithm to this predict ancestors of the future and further fitness. And since you've secretly stored fitness of all these sequences from your simulation, you can try to see how well this actually worked. And what you typically get is that from a static snapshot of sequences, you can infer fitness with about that sensitivity. So here I plot sort of inferred fitness versus true fitness, and you see that there's a fair rank correlation here which is noisy, but sort of picks up sort of some substantial chunk of the variance. This inference gets better the higher the mutation rate, which is another way of saying that the smaller the little increments in fitness that we do, the better the fitness prediction ends up being, which is expected since our model ultimately assumed that we have sort of small incremental infinitesimal increments in fitness, and that this process of changing fitness is sort of more of a diffusive one than a jumpy one. So there's another measure which is more relevant for the influenza question that we use to validate this with. For every such pair of past and present, we asked how close is this the node with the highest predicted fitness to the future. And this is shown here on the left, where we have this predicted distance on the y-axis and the post hoc optimal distance on the x-axis, and it's calculated as follows. So we look at the distance between our prediction and the average genotype in the future. We compare that to the average distance between these two strains, which means that the dashed line here is basically randomly picking. If you were randomly picking nodes from one season as representative of the future, you'd end up here. And another thing that we compared to is the optimal, and the optimal is actually shown here on the y-axis. So if you end up on this at the x-axis, if you end up on the diagonal, that means your predicted fitness, predicted node was actually the optimal choice. You could not have done any better than you did. And if you're somewhere in between these two lines, that's kind of it's a useful prediction, but it's not optimal. And when you see that on simulated data, in the majority of the places, you make a pretty decent prediction, and there's a minority where you're going wrong, but that's because evolution is a stochastic process, and this can obviously happen.
Yeah, so I would like to ask questions. So I'm trying to understand for myself why there is nothing on the bottom on the right triangle.
Yes.
What would those points mean?
Those would be predictions that are better than the optimal possible choice.
I see. Yeah,
so those can't exist.
Right. Yeah. Okay. Good. Yeah. Thanks.
A follow-up question. So, Richard, I mean, you mentioned this is a stochastic process, so you will. It's hard to always get it right. But how satisfied should you be? I mean, do you have a sense of how satisfied you should be with this result, of how good you're being able to predict?
That's a very good question, and at the time we were obviously very satisfied with this. But we've learned that there are some caveats to this result. I'll come to that in a second. But it's true that I don't have a terribly good quantitative understanding of how good you should be. So we've done now some follow-up studies where we are basically been asking, and this is together with Trevor Bedford again, if you not only wanted to find the closest match, but actually had sort of wanted to predict the entire distribution on the tree. You can define sort of various distances, which are in this case we use sort of what's called the Earth mover distance, or it's otherwise known as Wasserstein metric. One could also do something like Kullback-Leibler divergence or things like that. But yeah, but you have a more sort of a better sense in sort of how much information you're gaining by making this prediction on the future population. And I have to admit, I don't really have a good answer for you here. I think there is a lot of scope for interesting bits. Sort of some information theoretic analysis of this has always been somewhere on the list of the things we wanted to do, but it hasn't really sort of come to fruition yet.
All right, thanks.
Anyway, sort of, I promised to compare this to influenza. So this is what the analysis back then showed, where we do this for some 20-ish years, where the line at 1 is again kind of a random pick. The dotted line down here is sort of the post hoc optimal, And you see that our results in quite a few years are sort of close to optimal. Then there's a couple of years in between, and especially in the beginning, there's very few sequences. There's not much to shake a stick at here. One doesn't do terribly well. But overall, one has sort of quite meaningful prediction here that sort of systematically picks out strains that are closer to the future than the random strain would be. And remarkably, there's actually no influenza-specific input here. It's just taking a tree and sort of running, calculating some sort of index on them, and could be done for cancer or whatever, or other pathogens, whatnot, right? And this, right around the same time, sort of Michael Lessig and Marta Lukscher, they sort of put together a model that was meant to do exactly the same thing, and it has a fairly similar, maybe slightly better performance. Only that they took a completely orthogonal approach in that they were sort of trying to explicitly model influenza evolution and its interaction with the human hosts, arriving at fairly similar predictions. And it's been a challenge to actually try to combine these two approaches, and it still is a challenge in many ways, because the information that we are teasing out doesn't seem to be orthogonally complementary, but it seems to overlap largely, despite coming via completely different routes. And sort of before I close, and that's sort of one of the last things that I wanted to do, is I wanted to discuss some more recent work that sort of postdoc Pierre Bava-Chalet has done in the lab, where we were sort of trying to get to the bottom of what is it actually that we can predict. So we clearly can pick things that are better than average, but as Chase already pointed out, sort of what is that even sort of the right metric, right? And what Pierre was asking is, if you condition on seeing a new variant, say a new amino acid mutation, and if you condition on this having started at some low frequency a while back and you now see it at 30%, where is it going? Is this on average going to continue to go up? Is it going to turn around and sort of go back down, or anything in between? And what are the distributional properties of this dynamics? So with flu, we now have a number of years that we can hopefully actually make answer this question. And the results were sort of very consistent, but not exactly what we'd expect. So if you do this for a whole bunch of flu trajectories, so these blue ones here, you can ignore them, they don't make much of a difference. They just happen to have jumped the interval and then crossed it on the way back. But essentially what you see is that all these trajectories, they started sort of quite recently, they come up, and then some fraction goes up, some fraction goes down, and the average is essentially flat. So the black line here is the average of all these trajectories. And what this means is that there's absolutely no inertia in this, right? On average, you go up as often as you go down, and the chance that you actually end up at the top here is actually 1 minus the frequency that you started out with, which is exactly what you'd expect if you were just sort of randomly, neutrally evolving this thing forward. Which certainly puzzled us initially, because it's also very clear that flu evolution is not neutral at all. It is an evolutionary process driven by rapid adaptation for antigenic change. But somehow this does not seem to be reflected in the long-term dynamics of individual amino acids frequencies, at least once we see them at macroscopic proportions. So, Pierre then sort of went on a little bit more and sort of tried to, slice these data in all sorts of different ways. And there's very few indicators that you can use to predict whether a particular mutation is going to fix or not. And one of them is actually how often you see this variant showing up on the tree, like how many recurrent mutations, how often these mutations happens independently. So, but coming back to the dynamics a little bit, so here we show sort of, these the little bit weird plots. We make some proper histograms now, but this is a bit of a hangover. So if you if you just track the distribution of frequency trajectories, which were initially at about 30% through time. So, so that's kind of your initial condition. After 30 days, 60 days, 3 months, 1 year, and 3 years, you see sort of you see a pattern, no, 2 years, sorry. You see a pattern like this, where this is sort of gradually broadening, but after about, half a year to a year, there's essentially no information left where you are. It's just, you're sort of uniformly distributed with some fraction having fixed and the larger part having gone away. So Pierre then asked, how comes this, the LBI actually seems to be, so this fitness index that we devised, how comes this LBI seems to be predictive? And he essentially discovered something which is fairly obvious in retrospect, that the LBI has the tendency to pick out the base of fairly big clades, fairly big groups of this tree. So the LBI is big when things branch. That means it sort of tends to be maximal in sort of tight local clusters. Okay. And these tight local clusters are closer to the future than average. Because if you just look at the average Hamming distance between two populations, you get this blue line here as a function of time. And if you just take the consensus sequence of the entire bloody population, you get the green line. So the LBI is essentially a way to calculate the consensus of the population. And in many cases, actually the consensus sequence and the max LBI sequence are exactly identical. So we are sort of currently sort of trying to understand whether there is sort of, in addition to that, some kind of a signal of fitness, and trying to put this, to combine, reconcile these notions of something that looks like is just, picking out a consensus sequence, and on the one hand, and on the other hand, sort of something that is clearly driven by adaptation, how we can reconcile these two different views. There's something that's ongoing at the moment, but I thought it would be still good to include this here, just to show that, yeah, flu might not be, despite its initial appearance, it might not really play by the rules of these rapid adaptation models, which model the population as one wave that is constantly moving towards higher fitness.
However, may I interrupt? So I think it's super exciting, but we are sort of passing 6. So maybe if it's okay if you still have a few minutes to wrap up.
Yeah. So my talk's actually over.
Oh, perfect. Well, perfect.
So I'm ready to do the acknowledgements. So yeah, much of this influenza work that I told you about has been sort of a long-standing collaboration with Boris Freeman. The theoretical work on the Bolthausen-Slipman coalescent has mainly been done with Oskar Halecek. And for the prediction, also Flu and Colin Russell, who is now in Amsterdam, and Trevor Bedford have been collaborators over the years, as well as sort of Pierre Barra, who is now a postdoc in the lab and sort of investigating these more empirical aspects. And with this, yeah, this is all I wanted to tell you. I'm sort of happy to take further questions if there are any. And yeah, thank you for your attention.
Yeah, thanks. Are there any more questions? Yeah, thanks. That was very, very interesting. Thanks. Are there more questions maybe from our more junior people that are still around?
I'm pretty junior. I could ask a question. So yeah, it's pretty striking that this LBI quantity, predicts, has such predictive value. But if I understood it correctly, it's sort of defined with a radius, right? Which I guess is kind of a free parameter. Is that right?
Well, it's a free parameter in some sense. So we have a theoretical expectation for what it should be. Mm-hmm. It should be something like the coalescence time divided by sort of the log population size, something like that. So essentially, how many standard deviations out you are in the fitness distribution. For the flu predictions, we essentially just determined the the radius parameter of the LBI in simulation and just used that 1-to-1 for flu. So there wasn't a free parameter in that case, because we pinned that one down by simulations. But in principle, we also have a theoretical expectation for what it should be, and that sort of matches reasonably well. But it's fairly insensitive, which kind of in retrospect makes sense, given that it's, if it's some sort of a cluster and consensus detector, that of course has a scale on which you want to detect these clusters, but there's not like a gazillion clusters in these trees, right? There's typically sort of a handful of clusters, and the dynamic range there is not enormous.
I see. Interesting. Thanks.
Yeah, I think thank you very much. I think we should leave it at this.
Can I have one more question?
Who said?
Can I have one more question? If
yeah,
there's
of course.
time, of course. So thanks a lot for a great talk. I have a question on a slightly different topic. You showed this very nice phylogenetic trees, but could you please comment if there are some trends that can be linked to these trees in terms of the symptoms, severeness of the disease, incubation times, or something like that?
No, I don't think there's any information on these matters in these overall structures of the phylogenetic tree. So there might be specific amino acids, which of course show up on the tree, but there's no. I don't. Unless you already know the mutation to look out for, I don't think there's any meaningful way to tell it from the tree.
Okay, thanks.
All right.
Maybe one
more
last
question. Yeah, I've given up on being a tough chair, so go for it. I think everyone who wants to leave is gone probably.
Okay, so my question would be like, if there's actually, if you can predict essentially, can you use this maybe for perfecting a vaccine or something like this for the coming years?
Yes, so that is exactly the objective of all of this, right? So we are now sort of sitting on the biannual, so twice annual meetings of the WHO vaccine selection committees, and we do sort of an analysis of all their data, sort of trying to make predictions of what is circulating in future seasons.
Yeah. Cool.
One related question on that, Richard. I mean, you mentioned that flu seemed to defy some of the basic rules of the population dynamics model. Do you have a sense of what exactly is flu? How is flu really different from what you're assuming? Which assumption is really breaking down? And do you have a sense of how you can fix that or accommodate that in the model?
Yeah, so my current working hypothesis here is that we are, that it's mostly in the human population, that there is just a lot of diversity in the human population as how they recognize flu, and that any given adaptation in flu doesn't allow the virus to uniformly escape. It only escapes you in sort of some fractions, and they don't sort of overlap. And by that, it's essentially that sort of adaptive mutations are adaptive for some time, but they have an expiry date. Once they sort of run out of their fuel, they're essentially neutral markers.
So I had a very related question, I think, right? And you touched upon this. So to what extent would it be become important to take into account the interaction with the host, right? So in your current models, there's no explicit description of that. And so how important would that be?
Yeah, so I guess it's certainly important. The main question is whether there is a good statistical way of accounting for it, or whether you have to be explicit, right? And if you have to be explicit, sort of what is the data you base it on? There is sort of human serology where people, you take blood from humans and measure sort of antibody titers against panels of different viruses. So that gives you some idea of how of the diversity of sort of human immune responses against the diversity of viruses. So you have some sort of big matrix of recognition, and that would be certainly an ideal data set for that. it is not clear that we'll have that data anytime soon, because this, at least currently, the way it's done currently is quite labor intensive. So if there was a nice sort of high throughput way of doing this, that could potentially improve the way we predict and design these flu vaccines quite a bit.
Can I just interrupt? So I'm very excited about this discussion, but I have to leave. So I'm going to leave. Sorry, I have also other responsibilities. So I just want to thank both Richard and Kristina for really an amazing discussion. Please go on without me, but I really enjoyed this, but I have to leave. I have to feed my kids. So yeah, thank you very much.
Yeah, well, thanks for having us.
Yeah, good.
Yeah, thanks, Richard. That was a great lecture.
Yeah, it was very nice.
Yeah.
So I have another question, maybe. So if I get the flu, right, and my immune system fights this infection off, so how likely is it then be that if I will get infected, let's say next year, my immune system will be able to then combat that new virus, right? So in a year, this virus has mutated, and my immune system has responded to this strain of this previous year. So how broad is the defense? So is the defense so broad that it also responds to the new virus this year?
So in general, it's broad enough to protect you for several years. It depends, of course, a little bit. If you're unlucky enough that your dominant response is against the patch of the flu, which mutates the next year, then you might be in tough luck. But typically, these responses are somewhat diverse and polyclonal, and you'd expect to be protected for a while. That said, there's 4 different lineages of flu, and there is very little cross protection between them. So, people have studied this kind of the cross protection, and it is many years.
Actually, a question going back to the COVID or rather SARS-CoV-2 debate. I'm asked a lot from from friends and relatives about this topic, because I have some immunological knowledge, let's say. And one question that always keeps popping up that you would be much better in a better position to answer, I think, is if or not the virus is now mutating towards a, let's say, slightly more benevolent kind of state, given that most endemic viruses don't kill quite as much as pandemic viruses. So. Yeah,
I don't think there's any such evidence.
Yeah, okay, I say the same, but it's nice to hear.
And I also don't think that the argument is terribly convincing. All of us have had, we don't know what would happen if a completely new flu virus or another coronavirus entered a completely susceptible population. So the difference with these endemic viruses is that if you're 80-year-old, you probably have seen them multiple times in the year and you have pre-existing immunity, right? So against SARS-CoV-2, you don't have that, and this is probably what determines the severity of the disease much more than sort of some evolutionary game of the virus.
I see. So the notion that sort of viruses tend to evolve towards sort of less lethal but more transmissible, is this kind of a myth? I've heard this as well.
Well, they tend to evolve towards more transmissible, but it's not necessarily that this has anything to do with less severe. So over long time scales, this has been observed, but you could transmit more by staying asymptomatic for longer, or you could transmit more by making people cough more. So I don't think there is sort of a clear notion that this is one way or the other, right?
Yeah.
But would you say there's an exception for Ebola and viruses like this, where you essentially die really fast without medical help, for example? That there it may be true that they cannot really survive in a population because of that?
Well, there's many viruses that don't transmit terribly well, right? Sort of I think this has been studied in quite some detail in HIV, where there's sort of it's a long asymptomatic period, right? And in that asymptomatic period, the higher the viral titer, the higher the rate of transmission. But the higher the viral titer, the shorter the time to AIDS, right? So they are sort of conflicting objectives if you wanted to maximize transmission. At the same time, the virus evolving in the individual has, is sort of short-sighted, right? Sort of has no notion of, trying to transmit. It's only sort of happening at sort of a higher space and time level where you could see this sort of selection for lower viral load within an individual. This has been studied there quite a bit. There's some evidence that this is happening. It's not definitive, but generally I would say that this is sort of an attractive, an intuitive theoretical idea for which empirical evidence is scarce.
I see. Nice. I also have kind of an elementary question about these classes of models, right? That where, you have this kind of traveling wave of the fitness distribution. I guess that goes back to Fisher, originally, I guess, but here you also have kind of mutation selection balance, right? But in general, still the idea is that the fitness keeps increasing over time. Is that right?
Well, no. You always view this in a changing environment, right? It's not that things get indefinitely fitter, but that you have everybody else in the environment also changing, and this somehow sort of keeping a constant rate of replication.
I see.
But if the fitness wave is sort of portraying diversity in the population, which is always measured in this respect to some chemical potential, which sort of keeps up with the population.
I see. So across time, if you go back in time, you come from the fittest end, but it doesn't mean the whole thing's kind of shifting.
Yeah, you sort of look at the entire thing in some co-moving frame.
I see. Yeah, interesting.
All right, I think this is concluding the discussion if there are no further burning questions. So I'd like to thank both Kristina and Richard again. Amazing talks, and thanks for making this work so well.
Yeah, you're welcome. Goodbye.
Thank you.
Goodbye.
Thanks,
Have a
Kristina.
good evening.
Kristina, thanks, Richard.